Thank you, Frank. And thanks, everybody. I'm glad to be here sharing the research that we're doing to utilize microscopy images in order to search for cures for diseases and for, how biology works. My name is Juan Caicedo. I am a mortgage investigator and an assistant professor here at the University of Wisconsin-Madison. So I'm going to be talking about modeling cellular morphology using machine learning. So it turns out as the structure of the cell or the way we see the cells under the microscope tells us a lot about how biology works, how cells interact with each other, and what are the processes that cells undergo every second and every minute in all living organisms. In the screen, we can see images, example images of the cell cycle stages. That is a very typical phenotype that biologists like to look at because that tells a lot about how cells are reproducing and how we can see that under the microscope. But it's not just cell cycle. There are many other responses that cells can exhibit as part of their interaction with the environment. So if you have cells in a dish and you apply treatments or like chemical compounds or maybe even genetic engineering, if you like, you're going to see how the cell structure changes under the microscope as well. And that information can be quantified. It was back in the day when biologists looked at the microscope to decide, what's going on with the cell. But there are so many things that happen at the cellular level that we can no longer keep up by just looking at the microscope. We need to collect images and we need to automate the classification of those events that happen at the cellular level. And here is one specific experiment that people ran back in 2012. So they were studying leukemia. And what they wanted to do is to identify one chemical compound that was able to transform sick cells into a more healthy state. And the phenotype that they identified as useful was cell size. So in this case, we see control cells. Cells here are sick. And these ones are kind of healthy cells. And they are under this specific compound. So this is a chemical compound that was applied to the cells. And you can see the difference in size. It's just, the major phenotype that biologists want to recover in this specific experiment. Now, the original question was, among thousands of chemicals that you can use in order to produce this type of effect, which one actually works? And that's the biological experiments that people have to do in this type of research. So they finally identified this one as being the most effective by quantifying the total amount of DNA, which is the area of the cells in these images. And with that, we have a first type of a potential clinical trial for this specific compound in order to treat leukemia. This is a very nice example for several reasons. It's easy to visualize, like any person that can look under the microscope can tell whether the cell size is growing, but nobody is going to have the patience to do that manually and every time check under the microscope, because there are, again, thousands of potential chemicals that you could use, and we don't know a priori which one is going to work. The second interesting thing about this experiment is that visual variations in images tells us a lot about how cells are responding, but cell size is only one of the potential ways in which cells can respond to treatments. There are many other ways. So cell size is the easiest, and there are more complex reactions that cells can exhibit as part of any type of biological intervention. So in order to automatize this type of biological experimentation with microscopes, we developed a workflow which is called image-based cell profiling, in which we take images of cells and we transform those into data that can be used to strategize and decide what type of treatment is effective and which one is not. The way this workflow works is pretty much like this. Imagine that we have one set of cells that are healthy again, and then we have the sick version of those cells. And then you have another experiment that exhibits how the cells, the sick cells, respond to one specific compound or one specific treatment. So we have three conditions in this cartoon example. What do we do with the images? Biologists spend a long time preparing, the cells in order to be imaged. And after we collect the images, the first thing that we do is cell segmentation. And cell segmentation is the process of identifying where cells are located in those images, so we can then make decisions at the single cell level. This is similar to face detection, if you like if we are doing experiments with people and we collect thousands of images from social networks, the first thing that someone would like to do is identify where we can find people. So in this case, we want to identify where are the cells in one biological experiment. After doing this, we can create matrices that characterize the structure of every single cell in the experiment. So these matrices have single cells in the rows, and then a bunch of properties of those cells in the columns. And that is the process of feature extraction. So we take every single cell and we compute a bunch of properties or characteristics that define things like cell size and also the amount of colors that the cell has or the textures and so on. So many other properties that we can observe in the cells are collected in these matrices. So we can manipulate the data and then we can do treatment analysis. We also compute statistics of how a population of cells responds to one specific treatment. And that information is what we finally use in order to decide which treatment is useful or not. So this is the general workflow of image-based profiling, and it's widely used nowadays in high-throughput biological experiments in pharmaceutical companies and many other types of laboratories. And the take-home message here is that we can use imaging to inform biomedical research in many different ways. So drug discovery is one of the applications in which we can collect images of cells and tell whether the cells are responding in the way that we intend to have a response. But we can also do functional genomics or cancer research, all based on images. The only thing that biologists have to do is to prepare a high-quality sample, which is under the microscope, and then collect images out of that. And turns out there are robots that can do that, and many biological labs have that automated as well. So creating the samples is becoming easier and easier. That generates a bunch of images that, again, nobody is going to look at manually, and we need computational methods in order to transform all that amount of images, which is, like observing the microscopic universe and what's going on with the cells, and then interpret what happens at the cellular level in order to tackle some of the most pressing biological experiments of our time. So I'm going to describe a couple experiments in which we have collaborated with CHTC in order to advance the state of the art in those, in two major image analysis workflows. The first one is cell segmentation. And in cell segmentation, the problem, as I mentioned before, is we have an image like this, and we need to label every single cell, every single pixel in the image, indicating what cell the pixel belongs to. This is kind of a coloring problem, if you want, in which we are going to assign the same identifier to one specific object. And this is a relatively easy problem for images like this. However, when the cell structure becomes more complex, we're going to need more powerful types of algorithms, more specifically, the deep learning type of algorithms that are able to recognize more complex patterns. And the experiment that we were interested on recently is not about identifying cells, which is, or the nucleus of the cell, which is what we see here. We wanted to detect micronuclei, and micronuclei are very tiny objects in this image, and they are, labeled with red boxes. It's very hard to observe under the microscope as well, even for humans. And sometimes we can confuse it with noise because those images can be sometimes noisy, and the micronuclei are very, small dots in the image. So here we have an example of an input image. This is just a piece of the original image, and the manual annotation that an expert biology assigned to where the micronucleus is observed. So as you can see, it's kind of a hard type of pattern, and this requires more computational capacity to, process the context, understand the image, and produce an annotation, which is similar to what the experts produce. So in this context, what we wanted to try, given the complexity of this pattern, was a vision transformer network. And if you're following the machine learning, trends, transformer networks are one of the most powerful machine learning algorithms that we currently have. And they are behind the GPT, for instance, type of models. So there is a version that we can use for images, which is called vision transformers. And the way it works is it takes an image and then splits the image into the so-called tokens, which are useful for creating a sequence. In this context, it's not a sequence of words, it's a sequence of little patches that come from the original image. And then the transformer layers are going to analyze the relationships between all those tokens with the so-called self-attention layers to figure out what are, how they relate to each other and when a certain piece of the image makes sense for the prediction task. With the output tokens, we're then going to reconstruct an image that only has annotations in the dots that correspond to micronuclei. So this is kind of a computationally expensive algorithm, and it requires GPU acceleration. Our experiments require images like this. In total, they are like 3,000 by 3,000 pixels. The micronuclei are very tiny, and we have to look at a higher magnification in order to see them. In fact, we only had 18 manually annotated images. So this, is a computationally expensive problem, but it's not a big data problem, if you want. And the reason is annotating those images takes a long time. So biologists don't really want to, help us in that way. It's the opposite. Like we should be able to detect those patterns with minimal input. And so for that reason, we designed a leave one out cross-validation experiment in which out of the 18 manually annotated images, which took, about a month or so for the experts to check and verify where those tiny objects appear. So we're going to train with 17 of them, and we're going to leave one out in order to do the entire experiment. So one experiment actually has to repeat 18 times this procedure. And for each of the large images, we're not going to run all of the analysis in the full image. We're going to do it in a tiling type of pattern. So there is a lot of repetition here. And of course, exploring hyperparameters is also something that takes time. We need to calibrate certain parameters in the machine learning algorithm. We also need to calibrate certain parameters in the architecture and so on. So there is a lot of experiments that are required in order to do this. And here are some of the results that we got by running this algorithm. First of all, there was a baseline model that was reported in the literature called MN Finder for micronucleus finder. And it had an 80% precision and 42% recall out of the box. So if you use that type of solution, this is what you can get. In this study, they really wanted, higher precision and higher recall. So that was the primary challenge. In order to explore the possible combinations of model configuration, we had to run multiple experiments, and each experiment has to be repeated 18 times in order to check the leave one out. So here are some of the experiments, and these are kind of highlight experiments that show how the progression of those experiments went. I'm not going to go into the details of what all those configurations mean. It's just, the challenge of trying to beat the baseline either in precision or recall, but we couldn't do it in both at the same time. So it required more and more experiments and different configurations. And we were, so excited to be able to repeat those experiments quickly using the CHTC infrastructure until finally we managed to find, one configuration that was matching the precision of the original experiment, but actually making the recall double what it used to be. And this is the type of standard that the biologists that we're working with were expecting from this type of algorithm. So after, a hyperparameter search that took us about 2 months to complete, we were able to, beat the state of the art by a large margin. I think I cannot stress enough the importance of having access to, high throughput computing for this type of experiments. Most of the time, we have to wait until the experiment ends in order to get feedback and think again about what needs to be optimized in order to get a higher precision or higher recall. If that wait time is too long, the grad student is going to, be bored, and then they're not going to have sufficient ideas in order to push the problem forward. But with the infrastructure that we had access to, we were able to run experiments basically daily and think about, in what way we can push the experiment more and more. So this was what we were able to do for cell segmentation. And now I'm going to describe one experiment that is still ongoing for cell phenotyping. Cell phenotyping is the second part of the problem. If you remember, in our workflow, we started with cell segmentation, and then we do cell phenotyping or feature extraction, which actually aims to answer the question of how different two phenotypes are when we have one treatment or another. So for cell phenotyping, our goal or our challenge is to create a generalist model for bioimage analysis that can help us answer the question of, is one treatment working or not, more quickly. As I mentioned before, there are automated robots that can accelerate the pace of biological experiments, but now the bottleneck is the data analysis. What do we do with so many images? What type of machine learning algorithm we have to use? How long it takes to train it, to refine it, and so on. So what we want to do is to actually create a pre-trained model that can be used for any biological experiment. But turns out biological experiments may be very diverse as well in terms of the technical configuration. So here you can see that the typical natural image has only 3 channels, RGB, and that's kind of the standard. It's easy to pre-train a model when you have standardized images. But in biology, we have images of high contents screening that has 3 channels. The Human Protein Atlas is an example of a 4-channel panel. There are cell painting panels that have 5 or more channels, and biologists get creative all the time about how to put together images that can reveal more and more interesting patterns. With that type of variation, we really need to, get creative both in the machine learning side and also in how we can train a more generic model that can handle all that variation. And for that reason, we are investigating the pre-training of models at large scale. First of all, we are going to collect a bunch of diverse images that have been reported in the literature already, and they are deposited in repositories where people share the results of their biological experiments. There are many of those, and we started collecting images from those repositories. So the second thing that we are doing is creating machine learning algorithms that can be run at high throughput, in high throughput computing. And many of the modern machine learning algorithms can be parallelized, and that's why GPUs are so useful. But we also, have limitations in the number of GPUs that we can use. And there are many other ways in which we could parallelize the training of those models. And that's something that we would like to investigate as well. So ultimately, if we are able to combine large-scale image analysis with high-throughput computing, we hope to be able to provide, tools for image-based discoveries, because imaging gives us a lot of information about biological activity in cells and in tissues as well. So part of the progress that we have in this part of the study involves collecting images. So there is a bioimage archive repository where we collected 30 studies altogether, and it's 10 terabytes of data. We also use the image data repository, and we collected 70 studies from this repository for a total of 75 terabytes of data. And there is a consortium of image-based biological experiments, which is called JUMP. And that one has 12 laboratories contributing images constantly. And we collected 120 terabytes from that repository as well. We are trying to collect as much diversity as possible, because we know that in order to serve many labs, or in order to serve different types of biological experiments, we need to sample from as much diversity as possible. And for that, we need large-scale computation as well. With all that data that we have collected, and it requires multiple rounds of pre-processing as well, all that is done. And the challenge that we are facing now is, how can we train a transformer network using all of those images that we have collected, using the high throughput infrastructure that we have? One of the challenges that we currently have is how to communicate machines in a high throughput environment. The infrastructure that we use is great for independent type of experiments, but when we need to synchronize in order to train a single model, there might be some challenges that we could overcome. And that's, I guess, one question for the entire community. For now, we are sticking to the simple paradigm of training a model in a single server capacity and run it multiple times so we can check hyperparameters and so on. That is still ongoing work, and we hope that by training a model in a self-supervised way, we could hopefully recapitulate many biological studies. So this is part of our validation strategy. All of the studies that we collected in the previous couple of slides come from papers that biologists have published as well. So those papers report observations that the original scientists have made in those data sets. And what we're doing is collecting all that information and making sure that with a single model that we train, we are able to also observe those original findings that they reported in the papers. And the evaluation is also going to take a lot of compute power, but we're confident that we can run this in parallel in independent studies in the CHTC infrastructure. So with that, I have a summary here. First of all, imaging in biology is a powerful tool for observing what cells are doing and what's the response of cells to different types of treatments. Machine learning helps us identify a lot of interesting patterns that can be useful for biomedical research. And we are interested in creating models that can generalize across many types of biological experiments, and that would have a lot of applications in biomedical research as well. So with that, I want to thank the CHTC team, Brian, Justin, they are our heroes, and the Mortgage Institute, and many of our collaborators that provide us with images for this type of studies, as well as the members of my lab. Thank you so much.